Recording in progress.
Hello, everyone. We're just gonna wait a couple of minutes to begin our presentation just to give everyone a chance to join. Thank you. Okay, so let's begin our webinar. Hello again, everyone. Welcome to today's webinar. I am Marisa Fraser Morera with Pion, and I will be moderating today's event. This webinar is titled Advancements in Hydrogel Formats to Model Potential Drug Interactions Occurring at the Subcutaneous Injection Site, and will be presented by Randy Mersney, Professor of the Department of Life Sciences at the University of Bath, Kate Gridley, Research Assistant, also at the University of Bath, and Connor Gomes, our Development Scientist at Pion. Now, before I hand things over to our presenters, I would just like to briefly cover a few housekeeping details. We do want this webinar to be interactive and encourage you to contribute to its success by asking questions at any time during the presentation through our Q&A box on your screen. Any questions that we don't get to today will be answered by email after the event. If you need technical assistance during the webinar, please type your query into the chat box and send it to the moderator. We'll do our best to help you during the live session. I also want to mention that a recorded version of this webinar will be available on our website within 24 hours. And also an email with a link to the recording will be sent to all registrants as well. So now, without further ado, I will hand things over to Connor to take us through the presentation. Thank you.
Thank you, Marissa. And obviously, thank you, Randy and Kate, for being here with us. And thank you, everyone, for your attention. To reiterate Marissa's message, today we're going to be talking about the advancements in hydrogel formats to model potential drug interactions occurring at the subcutaneous injection site. This is sort of a review on a paper we recently published in the International Journal of Pharmaceutics and Biopharmaceutics. Today with me is Randy Mersney, who again is a professor at the University of Bath. He's also internationally recognized in drug delivery. He has election of the president of the Controlled Release Society and has organized a Gordon Conference on drug delivery, and is also the CSO of Applied Molecular Transport, a biotech company in the Bay Area in San Francisco. Lastly, Randy was also selected as a Medicine Maker 100 Powerlist for 2015 and 2016. And Kate Gridley works as his research assistant in the Mersney Laboratory within the Department of Life Sciences. And I'm Connor Gomes again. I'm the lead developer on the Scissor instrument here at Pion, where I've been for 3 to 4 years after receiving my doctorate in materials chemistry from Northeastern University. So to begin, today we are going to review the Scissor N3 platform. Randy will give a background on the development of the platform and its application in these spaces. And we'll go over the analytical methodologies that are used in the studies that we're presenting here today. We'll review the artificial extracellular matrices that we develop here at Pion and that Randy's developed previously for use inside the Scissor platform for the assay and development, for the assay of clinically relevant formulations. And we'll go through the specifics of how we used a whole bunch of different commercially available and our own formulated molecules to assay within these different artificial extracellular matrices to demonstrate the capabilities of each. So I'll hand it over to Randy for him to describe how Scissor came to be.
Thanks very much, Connor. I hope everybody can hear me and that my mute is correctly off. And thank you, Marissa, for setting this all up. So I'm going to go through just a couple of slides in background and kind of high levels. The slides, I think, will be available, and the content of the slides will be available through the recorded version of this. So you can go back and look at the specifics. There's maybe more information that's needed, but I really just want to highlight a few points about the progress of how we got to where we are to date and the rationale behind it. And it comes from the earliest stages of when biopharmaceuticals were started. The first proteins that were being generated upon injection into the various preclinical animal models, it was clear that nothing really made sense, that nothing modeled the human subcu site or the hypodermis for most of these proteins. And it was sort of a gamble in terms of how to formulate these and how to evaluate them and how to understand what might be expected when entering the clinic. And there was a big concern with that because the rationale is you could go into a very expensive early clinical question and either get no exposure at all or much higher exposure than you expected. And in both cases, one was very sad and underwhelming for the people there. But the second one was actually dangerous, that you could actually put more of an active agent into a patient than you were expecting. And so this was really something that was important. And we started looking at thinking about this. And if you contemplate how the subQ injection site materials are evaluated, there was really no criteria. I know we were looking at other molecules that were going in for the pulmonary route or other applications like on the skin. And under those conditions, there were criteria each time that you made these materials to kind of get a sense for the characteristics of them and stuff. And so we had the secondary concern that not only did we not know what was happening at the injection site in these different models, we didn't actually know what was happening to the material as well, because we had no basis for understanding how it might have been responding. And if there was lot to lot consistencies or variations in formulation components that might be affecting these things. So with that in mind, we kind of look to see what could be happening at the injection site and what might we think about. So next slide, Connor, please. And when we looked at the, do I do that? Thank you, Connor. So when you look at that, you can see that there's the injection site, the hypodermis in humans, is quite unique from a lot of other species. It's a place where we hold a lot of our water, our systemic water. And so there's this huge hydration capacity of it. And that structure that holds all that water is organizing fat for storage, for energy, but also a lot of activity in terms of movement of materials in and out. And so the picture on the left is a nice diagram, sort of the complexities of the various elements within that hypodermis. And we can model some of those things in an understanding of the format of those. And that's what this webinar is kind of about. And then if you look at the two images on the right, it gives you an explanation of kind of how people have misconceived this site. I think a lot of people think when you inject something in the hypodermis, there's a cavity or a cave of some kind that some material simply goes into and sits there. The reality is that it's a very compact site. It's a location, and so the material has to distribute through these matrices to actually fill the void, fill the site that you can deliver from. And so these things have the material that's injected has a lot of contact interaction with all those elements within the subQ site prior to release from the injection location. So next slide. So one of the things that we wanted to try to understand is what might be happening at this injection site and how it changes. Connor, if you can move the slide, please.
Yeah, sorry, Randy. The control is just kind of...
Okay, fine. All right, thank you. So if you think about what happens is we formulate these materials to be stable on a shelf for a couple of years. But the question is, what happens in the transition between the material leaving that needle and syringe that's drawn from that storage site, and then at the injection site and the interface between the materials that come in that formulation with all that matrix components, what could happen? And what you can envision is that the various elements that we usually use to formulate these proteins are potentially moving in their relationship with the organization they have within the formulation for that long-term stability. So you can imagine some of the agents now being attracted by components within the hypodermis and being pulled away from the protective function they might have. You can see changes in maybe aggregation content or state. You could see materials going from below a critical micellar concentration to now above critical micellar concentration in terms of their coordination around certain locations. So you can imagine a lot of things happening at that site, and we really had new tools to understand really what might be happening. And one of the most critical things that we had demonstrated early on was that most companies were simply taking their material that was sitting in a formulated solution in a vial, something, and they would simply just inject that into phosphate buffered saline. And it turns out that that was really a very artificial, inaccurate assessment of what might be happening to the protein, primarily because it is not very physiologic. And we know that when you move these materials from these formulations into that injection site, many times they could be passing through their isoelectric point, and these various elements could be happening because of that that's outlined in this slide. So we wanted to understand more about the bicarbonate-based buffer environment that the body uses to buffer this system, and the conditions that that the proteins might experience in that location. And so we set about trying to figure out how we could model that. So the next slide is to show that, in a very simple system, we're just taking a dialysis cassette. We could show that you could put material into that cassette in a large bucket, basically of material of fluid that sort of emulated the infinite sink of the body in a physiologic sense from what it was composed of. And we could watch the material within the injection site, that is within the dialysis cassette, and we could watch it also leave and monitor both of those parameters. We could monitor the pH and check the conditions of the things that are coming out. And in doing so, we could actually take advantage of two components here, which is the spectroscopic status of what was going on inside the dialysis cassette, actually looking at the protein and its condition in terms of solubility versus precipitation, and also the flux of materials leaving. So on the next slide, we modeled that chamber and looked to see if this chamber actually did that. And so the scissor then could be formulated with the idea of assessing these components. And we could show from our early studies where we used various materials within a cassette or that dialysis chamber, we could watch the material leaving that compartment, and we could monitor the pH of the components in it. And so this is just a composite of some of those early studies that showed that indeed we could follow the pH over time pretty accurately. We could watch the deconstruction of the materials that we used as our artificial injection site by adding something back to them, and would show that the change in release of material from that chamber was quite consistent with that. So we could show the modeling of the systems. We could show pressure changes within the compartment, kind of consistent with the idea of what might be happening when you add material to a place that doesn't have a cavity to hold it. It has to pressurize the system a bit. And we could look at the solubility of the environment around the injected site to see that we can make models where we could look visually, optically clear through the material to actually evaluate what's going on. And so that's some cuvettes in the bottom there showing the condition of the components there with collagen or hyaluronic acids and components. But most importantly, what this system gave us the chance for was the idea of watching individual materials leaving injection sites and what the condition of them were within the injection site and what condition they were when they came out. And so by taking an insulin solution initially, we could actually watch one of the stabilizing elements, metacresol, come out very, very rapidly from that injection site, and the protein coming out slower. And you could actually watch the protein sort of precipitate at that site as the creosol left, and then slowly redissolve and release over time as the pH of the system equilibrated from the formulation pH to the physiologic pH. And so this was a really nice first test showing that we could actually, based on the things that you're seeing in the rest of the slide, really follow what was going on inside the injection site, follow what was coming out of the formulation system, and then really understanding sort of what might be happening internal to the system. So we then went on and looked, the next slide, we'll talk about 4 quick antibodies that we first looked at to validate the system, monoclonal antibodies. And we really wanted to understand, how do we model those? The most critical part of what we were trying to do at this point was to correlate the materials that we got from clinical outcomes with what we saw in the scissor system. And that was important because from the very first slide I showed, we know that there's no animal model that perfectly recreates or predicts what happens in patients. And so we were trying to get materials, and we were lucky enough to get antibodies with clear, well-established information from the clinic, and we could correlate that with our system and sort of set it up to understand what parameters might be useful for us to predict or assess what was leaving from the injection site, and more importantly, what might be happening at material retained at the injection site. And so this early process allowed us to actually come up with a criteria or a system for the early chambers that we used, and to use that for a validating parameter, validating question that we could use then to say the scissor system might actually be used to assess other formulation materials. So about that time, we went into contract with Pion. The university set up a system with them, and on the next slide, we helped develop what is now the currently commercial scissor, which Pion has now taken forward and has really done the beautiful engineering to make sure this is a functional, rapid use system. And it's not the one that where it's the idea is that somebody can come up to it and just use the instrument as it is and get the kind of data that we were talking about a minute ago. And so with that, I'll turn the system back. I'll turn everything back over to Connor, and he can talk to you more about how the system is organized now and how we're taking it forward. Thank you.
Yes, thank you. So thank you, Randy, for the beautiful intro on scissor and how we have the current one developed today. As Randy mentioned, the image on the right shows our current scissor N3 platform, where inside each individual one of these chambers, you can run your own individual injection for formulation analysis. So within each one of these chambers, you'll have the cartridge with the artificial ECM that Randy mentioned previously, along with the bicarbonate buffer that's balancing pH and keeping everything biomimetic. You'll also have images being taken over time of the bolus formation and any formulation phenomena that happen thereafter injection. And we have pH and turbidity monitoring of the ECM itself, the artificial ECM, and the outer bicarbonate chamber. And so today we're going to focus a lot on the development of the artificial extracellular matrix that's found at the heart of the instrument. And so Randy describes the development of the largely glycosaminoglycan-based ECM, where you have these long-chain saccharides that are present in a buffered solution. And they stay optically transparent throughout an assay so that bolus formation and post-injection phenomenon can be seen quite clearly from either viewing it with a human eye or from the optical images that we're collecting over time within the current scissor platform. To expand on these GAG-based ECMs, we will present a extended release version of them, which has covalent crosslinks mixed up within these glycosaminoglycans to increase the structural stability and to extend the amount of time that the bolus and injectate is staying inside the simulated ECM environment, so that those post-injection phenomena can be viewed so that there can be some clinically relevance gathered. And lastly, to further expand on those versions, we're going to talk about the addition of biomolecules into these systems. When you inject into a subcutaneous space, you have solid and aqueous proteins all creating this complex matrix. We're going to talk about when you broaden the electrostatic activity of those samples, as well as adding in more positive molecules like collagen and other classes of enzymes. You can achieve different injection phenomena that can be viewed, and you can gather different information from the pharmacokinetic release curves. So to start, we're going to just look at the optical behavior of each and talk about the biomolecule inclusion into each of the non-crosslink and crosslink versions of the glycosaminoglycan-based artificial ECMs. So first, I want to direct your attention to the images that we can see on the right-hand side. those are all examples of collagen type 1, collagen type 2, collagen from human fibroblasts, and then finally chondroitin sulfate added to each of these artificial extracellular matrices. Just like the previous publications that Randy showed, and if anyone has ever made a material out and added collagen to it, as you add collagen to a aqueous environment around physiological pH, you get an increase in turbidity over time. What helps the turbidity early on is actually work done by the biomaterial design group headed by Dr. Leila Daravi out of Northeastern University, where these glycosaminoglycans can actually delay the assembly of a solid collagen construct that's supposed to spontaneously assemble at physiological pH. And these ECMs sort of take advantage of that, where we can increase collagen concentration slightly, where we don't have significant increases in turbidity, except for specific systems. And the observations that you can see on the right-hand side are supported by the graph on the left-hand side of optical density measurements, where as you increase the concentration of collagen, you have an increase of turbidity. One thing to note is that when the ECMs are in storage condition, and that's at 4 degrees Celsius, there was no appearance of any turbid aggregates only until thereafter. So yes, and there was no real difference between the optical appearance of a cross-linked, the XR version of these hydrogel formats, and the non-cross-linked version, which shows that the crosslinks that we're adding may not be significantly impacting the chemical reactivity, simply the structural characteristics. So next, we employed some rheological measurements to show the viscoelastic properties before and after an assay with the glycosaminoglycan-based non-cross-linked samples. We gathered 72 hours worth of incubation data. So this sample was placed inside of a scissor assay at functional physiological conditions, pH 7.4 and temp 34 degrees Celsius. And before and after that assay, we took these viscoelastic measurements on a sandblasted plate geometry with oscillating frequency due to the hydrogel nature. And you can see that after these 72 hours, from about 1 to 15 rads per second frequency, during that stress, we have sort of a conservation of some mechanical stability at the end of these 72 hours within a collagen-containing sample. And when you do not include those molecules, you have even more significant viscoelastic loss over time. This is most likely due to the fact that the partially positive collagen and the negative glycosaminoglycans are interacting together to sort of stabilize the matrix within the cartridge. When you add in covalent crosslinks to that system, there is no real difference between the collagen-containing and the non-collagen-containing samples. And those covalent crosslinks are able to conserve the mechanical properties of the system over 144 hours worth of assay. So yeah. And lastly, before I hand it over to Kate to talk about some even more injections, I want to just show the bolus formation characteristics of each of these ECMs. So the top row will show a primarily glycosaminoglycan hyaluronic acid-based ECM. And within this matrix, you see the bolus formation and then a homogeneous diffusion of a horizontal front from the top to the bottom of the cartridge, possibly due to the slight change in density over time, until complete dissolution out of the cartridge to form some clear or homogeneous cartridge with the outer chamber. When adding in collagen expressed from human fibroblasts, which will be primarily a mix of collagen type 1 and collagen type 3. You see similar or analogous bolus formation, but then a more homogeneous diffusion throughout the entire cartridge, more of a chromatographic diffusion or chromatographic method diffusion through that artificial ECM in all directions from that bolus. And then lastly, we see the extended release or cross-linked format. You can clearly see that the bolus formation is not as nice and spherical or elliptical as the uncross-linked versions. However, the bolus is sustained over a long period of time, which is the objective of the model, is that it's to hold your formulation within that reactive artificial extracellular matrix environment, so that you can see those post-injection phenomenons over a longer period of time. I apologize, these injections are of 70 kilodalton dextran molecules. And we used these and as well as a 4 kilodalton version analog to show the differences, the molecular weight correlated behavior within each of these ECM formats. And really quickly, every single format showed the correct release order, rank order of the lower molecular weight 4 kilodalton, releasing faster from the cartridge than the 70 kilodalton version. However, with the extended release version, you can see that complete release was not achieved, sort of hammering home that point that this hydrogel model is maybe more appropriate for viewing just the bolus interaction with the surrounding ECM environment, rather than tracking the pharmacokinetic behavior within the model and try and relate that to the clinical response. So with that, I'm going to hand this off to Kate so she can describe more of the injections of formulations that we carried out to understand these hydrogel models.
Thank you, Connor. Okay, so our next step was to examine the matrices with the small molecule drug, because we wanted to observe if passive diffusion is affected by the inclusion of the crosslinks and the collagen. So we used caffeine as a compound which is slightly polar and it's readily water soluble, and therefore it should theoretically be released rapidly from the cartridge. So hyaluronic acid and cross-linked hyaluronic acid in this data here showed extremely similar release in the first few hours, which demonstrates that although we've changed the mechanical properties of the hydrogel with the cross-linking, this hasn't impacted its chemical behavior in relation to this small molecule. As you can see, one of the replicates in green, thank you, Connor, exhibited a partial burst release upon injection. And although there are a few ways this can happen, the primary way is via reflux up the path of the needle. So this data would be a statistical outlier and wouldn't be valid to define the release characteristics of a material. However, we included this as a sort of learning element. So it's a behavior which can be experienced in scissor, and it's a common phenomenon when too high a surfactant concentration is used, as was shown in a previous PION webinar where they looked at the use of polysorbate 20 and 80, and I believe it was polysorbate 20 that had a higher rate of early release. So as I say, it's something to look out for, and it's potentially a formulation dependent phenomenon, and it's not unlike, that is to suggest it's not unlike would be observed in the clinic, i.e. if a formulation experiences this behavior in scissor, it may well experience the same thing in a clinical setting. And in blue, you can see the collagen assays. So the addition of collagen consistently resulted in a lag in the initial release, which suggests that the electrostatic interactions are occurring between the drug and the matrix with collagen, and that caffeine is actually complexing more with collagen at the site of the injection. If I could have the next slide, Connor, that'd be great, please. Thank you. So next, moving on to some commercially available formulations, we looked at rapid prandial and longer-acting basal insulins to further validate the correct rank order that we can achieve using our artificial extracellular matrices. And it's really reassuring that we note similar release profiles for Act Rapid. So these are the pastel colors on the left here. We noted really similar release profiles for Act Rapid within the first few hours for both of the hyaluronic acid-based matrices. So it's once again demonstrating that chemical similarities between the hyaluronic acid and cross-linked hyaluronic acid formats here, with a molecule that was designed to quickly diffuse through the matrix, being a rapid-acting insulin. You may note the seeming decrease in concentration of the Act Rapid with the collagen-containing hydrogel. That's in light blue. Thanks, Connor. And this consistent observation, i.e. it's quite repeatable, there's very tight standard deviations there, could be due to the relative instability of Act Rapid and its degradation. It seems to be collagen dependent, and it could be due to isoelectric precipitation, but we need more testing to validate this. So next, looking at the longer acting insulin Levemir, as expected, thanks. Same slide, sorry. As expected, the release of insulin basal is much slower relative to the act rapid, which makes perfect sense when comparing to the clinical data, where rapid insulin typically lasts for about 4 to 6 hours, whereas basal insulin has a duration of up to 24 hours. So we see the majority of rapid insulin released within the first few hours of the assay, and the majority of basal insulin within 20 to 24 hours, which is great. It's harder to differentiate in this long range graph because it's a very long time frame, and I'm asking you to look at the first portion. But analogous with the caffeine run, we saw previously, collagen does show a slight lag time in release. You may be able to see a little lag there, which, considering the rheological similarity between the two matrices, between collagen and non-collagen containing matrices, this suggests that there are more complex interactions occurring with the collagen matrix. So next, I wanted to look at the fact that with the cross-linked hydrogel, cross-linking massively reduces the release rate for basal insulin. In orange, we've got a gradual and really minimal release of basal insulin from the HA XR matrix, and this is largely as we'd expect for this combination. The cross-link matrix was designed to sustain a bolus within the cartridge for an extended period of time, which is most likely why we see retention of the material within the cartridge, on top of the fact that it's a formulation which is designed to release over a long period of time as well. So the cross-linked HA-based system reduces diffusivity, so it can be used to view, as Connor stated previously, it's possibly better to view post-injection behavior and stability and performance of the material in a stable bolus. This is the first time we're demonstrating using longer runs with scissor, so up to 80 hours here, and I think even more in some of the other assays that we showed. So we're really showing how the system can be used to monitor long-term injectables as well for longer periods with increased stability of both the API and the matrix. So overall, I was just going to say we're just really really reassured by the fact that all of our glycosaminoglycan-based matrices all showed the correct rank ordering. So we've had a good success there. If you could move on to the next slide now, thank you. Thanks. So next we wanted to look at denosumab. I'm sure as many of you are aware, subcutaneous administration of antibody therapies is increasingly popular route of delivery. So here we looked at the monoclonal antibody Prolia, which is used to prevent bone reabsorption and a variety of other similar conditions. So in the data here, just looking at the bigger graph initially, all the experiments showed similar release kinetics at the beginning of the assay, where initial release seemed to be a bit faster than the subsequent more gradual linear approach to the maximum. And that's as we'd expect, since Prolia is designed to act over a series of months in the clinic. And this is in concurrence with clinical data as well, where Prolia shows a bioavailability of about 61% following subcutaneous injections. So that fits in quite well with our data here. Now, the lower concentration of collagen had a slightly faster release to maximum, but both collagens in blue approached the same value of about 70% release, with both had excellent standard deviations here. You can see they're very tight. With collagen, we create an electrostatically diverse matrix with more potential interactions for the biomolecules with complex charge distributions to interact with, compared with the hyaluronic acid-based hydrogels alone. And once again, in orange, the cross-linked matrix allowed denosumab to be released and monitored more steadily. So I'd like to just point out very briefly that the sort of supra 100% release you see in green observed in hyaluronic acid in some of the assays. With multiple day experiments, we can sometimes see turnover of materials, and proteins can experience self-association and aggregation events. Sometimes, particularly the longer the duration, obviously, this can change over time, and it can create scattering events. And we suspect that in the HA hydrogel, the antibody and the hyaluronic acid was released over time, and the resulting complexes and aggregates can result in these spectroscopic anomalies. anomalies. And just wanted to say as well, in the cross-linked matrix, the steadier release of prolia suggests that the protein is complexing with hyaluronic acid. And it was since the cross-linked format, more HA was maintained within the cartridge. This could explain the difference in results between hyaluronic acid and cross-linked hyaluronic acid. So moving on to the images below, it's not labeled, but these came from an assay with 0.1, I believe, mg per mil of collagen, fibroblast collagen. Yep, that bottom one that Connor's pointing to right now. So we can see aggregation here within the cartridge from about 3 hours, it's difficult to see on here, up until about 30 hours, at which point it does start to dissipate. This also interestingly corresponds to the graph above, where the LED transmission average and also, well, it increases roughly when the aggregation seems to disappear, or the turbidity seems to disappear. At the same time, there's an increase in pH with the aggregation and then a drop once it's cleared. So biomolecules and monoclonal antibodies can degrade over time, but to determine whether this is caused by the formulation or if it's matrix dependent, we'd need to do some further studies and analysis. But in this tractable system, you can quite easily monitor more complex events. And yeah, that's about it. Thank you.
Thank you, Kate, very much. And continuing on that theme from Kate, we just have one more set of experiments to talk about before we conclude. But this is a commercially available formulation of bevacizumab, Avastin. And we looked at this formulation to try and track the collagen-specific interactions that can occur within the collagen-containing matrix versus matrices that do not have any biomolecules contained or any collagen contained within them. So bevacizumab targets and has affinity for VEGF, and the VEGF molecule has an affinity for collagen. So our experimental design was to try to build up the ECM components, where you start with your simple glycosaminoglycans and you build up and include each biomolecule, collagen and VEGF, and then inject the bevacizumab into each of those different models and see the response from each. So hopefully we can see the VEGF complexation to collagen and the bevacizumab complex to the VEGF to slow the release of each formulation with each iteration of these compositions. And so you can see that just to start out, when we add in those biomolecules, much like the collagen, you have an increase in the broadness of the electrostatic sort of chromatographic interactions that will happen in between the bolus and the membrane release into the outer chamber. And so the HA itself had the fast release, while the ball meal containing matrices slowed that release. With the inclusion of collagen and VEGF, which is the blue curve at the top, that was the fastest release curve. So there could be a concentration dependent interaction with VEGF, as the 10 microgram per mil VEGF had a lesser release. So there could be more complex electrostatic interactions going on and potentials being felt by the formulation that dictates its diffusion away from the bolus. That is not simply due to the fact that it has affinity for a specific biomolecule. So although these curves, we do all demonstrate the change in the pharmacokinetic response based on the addition of each of these components. However, to understand the specificity of a collagen affinity, we're going to need to run more experiments, possibly with something more specific, like just a collagen antibody, to show that action. Because although a lot of molecules have some sort of interaction with collagen, a specific one is very hard to find naturally in biology. So this is probably a continuing study. And with that, I just want to conclude that we showed that from the simple glycosaminoglycan-based ECMs, we can increase that complexity with some covalent crosslinks that do not significantly impact the chemical reactivity of the model, but just change the mechanical structure of it, or mechanical properties of it, so that bolus formation and post-injection phenomena can be viewed over a longer period of time. Biomolecules being included, specifically collagen within that ECM, those glycosaminoglycan formulations, solutions, excuse me, increase the mechanical stability, while also broadening the electrostatic interactions that happen within the cartridge to hopefully catalyze more post-injection phenomena that have more clinical relevance than simply the glycosaminoglycans. And so looking forward, our goal at Pion is always to establish that in vitro, in vivo relationship with the scissor assays. So over time with BAP and others, we are constantly looking for that relationship with either the inclusion of biomolecules or the advancement of the types of formulations that we assay within scissor. Because now that we've shown that we can include structurally or more labile biomolecules that are found in the in vivo environment, including more enzymatic processes or more active processes within the ECM using those biomolecules is of very much interest. And actually with this, we have a white paper coming out soon that shows we are able to conserve some of those enzymatic or inactive to active drug transformations that require a native biomolecule. And that white paper should be coming out soon to demonstrate the use for like prodrugs and stuff like that within scissor. And then lastly, we do want to get a really good handle on the impact collagen has on these post-injection phenomena and formulation behavior, because it seems like getting that specific interaction down is a key component to understanding the relationship between an in vitro assay containing collagen and an in vivo response. So with that, we want to thank again Randy and Kate for their continued and irreplaceable scientific contributions for our scientific collaborations. And if you want to read some of the papers or our most recent paper, I have some of them cited here. And with that, I want to just thank you for your attention. And if there are any questions, we will be answering them now. Once I can get the Q&A to pop up.
Are you able to access a Q&A?
Yes, I
panel?
am. Yep,
Okay.
I just got it.
Great.
So the first question, I understand that a 300 mil chamber has typically been used for scissor runs. Has there been an instance where the 60 mil chamber was utilized? If so, would we anticipate observing a similar release for both the 300 and 60 mil chambers? So what a great question. The standard assay is done with a 300 mil chamber. That 300 mil, hopefully I can go back and just so people know what we're referencing. So within the scissor design, we have the outer chamber that contains the bicarbonate buffer. You can see the 300 mil outer chamber here. There's a 60 mil chamber that is just of much lesser volume. And so the 60 mil chamber is used for molecules that, one, do not release a lot from the internal chamber, or two, are very hard to detect spectroscopically. So in a nutshell, the answer is concentration dependent for in situ analysis is the primary reason. The reason the standard assay is 300 mils is because it has a longer sustained sink condition with complete release of an injectate from the chamber. So with the 60 mil chamber, if you do employ that and you have bulk release from your ECM chamber, I don't believe sink conditions will be conserved over all systems. So if you have like a 10% release of a solution that goes into the outer chamber in the 60 mil, you're still going to conserve sink conditions because the concentration gradient is so large. But as soon as you start to get bulk release in that 60 mil chamber, the sink conditions, as well as the events you see in the outer chamber, start to change, because as you increase, especially with biomolecules, as you increase API or biologic API concentration, you increase the chance of aggregation and shear within the system from stirring. So those are the primary reasons why you would choose one over the other. Do the new matrices work for when using the rainbow for API quantification? Yes, both ECMs have been used for a lot of the injections that you saw today. So we know that the cross-linked version and the non-cross-linked version will not significantly impact at least the spectroscopic response. The only thing that I would mention is that if you do have something like a larger protein or a MAB, typically when they spend a lot of time in a stirring solution, they'll start to shear and aggregate themselves. In addition to that, the non-cross-linked version of the ECM will have more material turnover, and therefore more hyaluronic acid will exist in the outer chamber. This is fine, as that's how the subcutaneous space acts in the body. You get material turnover the whole time. That's how a lot of the bolus is leaving it circulated. But what this means is that any of the shear mediated aggregation or any of the just aggregation that's happening due to the bulk material properties within that outer solution, the aggregation will probably be increased with the non-cross-linked version, because you have extra long, like very, very large kilodalton, megadalton polysaccharide chains coming out of that cartridge too. And so that would be really the only difference in Rainbow. But actually there's methodology tricks you can play to remove that baseline elevation and aggregation sometimes, but they both work with Rainbow, and they both should work with any other offline analysis that you would have. All right. So I can yes, I can mention about the stability of each ECM. So the uncross-linked version of these ECMs, we typically have a standard assay of about 24 hours. Within 24 hours, you get a significant amount of material turnover, which again isn't inherently which is related to like what happens in the clinic. But what that means is like the stability of the matrix will only be valid within the time frame in which the material still exists inside the cartridge. With the XR, I showed a 1-week experiment previously with the viscoelastic curves, but we can assay over 1 week and still conserve the mechanical stability of the cross-linked models. Yeah. And I think, let's see. I think that's generally it. Let me go see if there are any more questions. So yeah, I guess if there are no more questions, I'll give it another 2 minutes, but if there aren't any more questions, we can
Connor, I see another question here.
Oh, okay, great.
How stable are these ECM formats?
Yes, that's the answer I just gave, but
Oh, you just gave that one. Okay.
there's another question here. Is there any concern that the method of cross-linking for the XR matrices conditions that are not representative of the subcutaneous space? What a fantastic question. So we did do the best we could with conserving the biomimetic processes that are present in an in vivo environment within our manufacturing processes for making the cross-link versions. So the bonds that we're including are very analogous to a peptide bond. So there's not any chemistries going on where we have like any excess catalyst that's present within the actual backbone chain that we're creating. It is an enzymatically catalyzed cross-link, and so we're not including any or any synthetic polymers or any synthetic materials within the manufacturing process. So we're pretty sure that all reactivities, and from the similarities we see with our own injections, we're pretty confident that we're not adding in any additional reactivity that wouldn't be seen in the sub-Q. So yeah. And then we have a question here. If the bolus diffuses, the surface area will increase. Will that affect absorption? Yes. The accessible surface area that is seen from a bolus is directly correlated with the rate at which the body can interact with that bolus. But there's also the play of concentration dependence reaction of that. So there's a lot of like concentration mediated processes in the body. And so I think that to answer, I'm just going to go back to your question and answer it specifically. As the bolus changes, the rate of absorption will be changing. And that's typically related to the pharmacokinetic response or the rate of release that you see in these in vitro assays. So yeah, a bigger bolus will be diffused differently than a smaller bolus. To answer that. In the case of the XR cross. Yeah, I think our objective is reproducible bolus formation. The bolus will take up there will be larger surface area available within the XR model because the void volume available for the bolus to take up volume inside that cartridge is lesser inside the crosslink versions, which means you're pushing a volume through all these different little tiny channels that are existing in the ECM rather than displacing bulk solution. So you're increasing the rate at which surface interactions will happen between a bolus and a component within the artificial extracellular matrix. But although you're expanding the surface area to increase those interactions, you're not going to affect absorption in the in vitro assay. So in an in vitro assay, in these examples in the XR, the accessible surface area is not correlated to the rate of release because the model that we'd be comparing it to, I believe this question is comparing it to like the normal ECM. The normal ECM, it would change the diffusion. In the XR, its job is to hold that bolus in. So I think even if you changed the volume or changed the rate of diffusion, it would still be conserved within that cartridge. But thank you. Good question. I can, all right. Oh, is the viscosity of the matrices similar to the subcutaneous space? So it's not a super straightforward question because it's been so difficult for scientists up until this point to get stable viscoelastic measurements of the subQ space. So we have specific things like other ECM spots like the vitreous humor and interstitial fluid. We have viscosity of those things because they can be collected as a bulk solution. But with the subQ space, there's such a mix of adipose tissue and tight spaces and that are sort of participating in the viscoelastic nature and in the mechanical nature of the ECM that it's very hard to like match that environment. We can say that we are of a viscosity that allows for the stable diffusion of formulations within an in vitro environment. But there is no direct number that we are trying to match with the viscoelastic properties that are found in the subQ space and the scissor instrument. I can bring up one slide again. Contact information. Yes. So, sorry, is scissor available for purchasing and how can I get a quote? Yeah, scissor is available for purchasing. You can contact Pine or email me directly. I'm so sorry I didn't put my, I'll actually just write it now that we are here, but I can't, I'm losing control. Anyways, you can purchase, you can contact Pine at any time. The scissor instrument is both available for purchase and we have analytical service agreements with the scissor instrument where more robust sort of or more complex ECMs have been developed for analytical service purposes. But yeah, the Pion instrument and it as an analytical service are both available for purchase collaboration, and we are open to all that communication and are available to talk about and discuss all the nitty-gritty about the scientific steps that need to take place. Is there a volume limit for injection? Within the assay, we've had injections sub 100 microliter and up to 0.5 ml to 1 ml. What I'll say is that the amount of the ECM will work a lot like a chromatographic method, and like you can think of like plates of interaction, where, excuse me, the larger the injection, the less interactions that can happen with the formulation in the ECM before it exits the cartridge. The recommended volume that I've been using for projects is around 250 microliters. That seems appropriate, where you can people, if needed, can still use a more clinically relevant volume, so you don't have to go so small. So you can still get up closer to the mil volumes that people normally inject, or on that magnitude. But really, that smaller bolus allows for more interaction of the ECM, which can help understand the post-injection phenomena that are happening. If you inject too much, like 0.5 ml, a lot of the potential energy that the API will field come from the bolus itself and its own formulation forces, rather than the simulated sum-Q interactions that we're trying to simulate within the scissor instrument. So volume limit, I'm recommending 250 microliters. If you want to do 0.5 ml, up to you.
Thanks, Connor. I think we've run out of time.
All right.
If we didn't get to your question, we will answer offline. I want to thank everyone for joining us today, and also thank you to our 3 presenters. We really appreciate your time and efforts in putting these together. As I mentioned, the presentation is recorded and will be available within 24 hours. If you have any questions, or for the person that wanted to know about the scissor and a quote, you can also go to our website, where you can request a quote there from the scissor page, or email us directly. And also do check our upcoming events, where you can sign up for future webinars, or look at all of the different conferences where we will be present. Thanks again, everyone. Have a wonderful day, and we hope to see you again.
Thank you,
Goodbye.
everyone.
Thank you.
Thank
Bye.
you.